Yes, good evening, Pooja. I'm sorry I was not able to post enough classes for you for the last week because we are pushed with some commitment over here. So let me start the class for you. So let us revise what we have seen in the last class. We saw about the various means of transport, where we have seen about active and passive transport and diffusion and facilitated diffusion and the means of transport. What are the various porins are there by which the molecule can go in and out of a cell by means of the plasma membrane. So passive transport is a means of transport, as I discussed with you. There's no need for any energy. So it can be a simple or it can be a facilitated, where energy is utilized in the form of ATP in case of active mechanism. For this, I gave you a mnemonics called as Nokia. Nothing to worry. Again, when I go with the active transport in depth, we can see something about Nokia. So, simple diffusion, and we have seen about the movement of molecules. The molecules are able to move along the concentration gradient, not against the concentration gradient. That is, from the higher concentration to a region of lower concentration, the molecules are able to move in case of diffusion because it is a passive transport. Then I've discussed about osmosis, and osmosis deals with the movement of solvent molecules. So a solution is made up of one solute and a solvent. The solute concentration will determine the tonicity, the concentration. Based on that, we have discussed what is hypo means less. Okay, it's less concentrated. That means the concentration of solute is less, solvent is more, high water concentration. And hypertonic is a one where we have more concentration of the solute and low concentration of water. The water molecules moves from hypotonic to hypertonic, that is higher concentration of water to a region of lower concentration of water, where tonicity stands for not for solvent, it stands for the solute. Then we have seen about what will occur in case of a cell, we take a plant cell, animal cell. When we dip in a normal water, that is in hypotonic solution, generally what happen, it will swell. But in case of animal cell, it will burst. Why it will burst? Because there is no cell wall. And in hypertonic solution, it will shrink. So these are the following topic that we have seen in the last class. Now we are going to see about a special mode of diffusion, and that diffusion is called as facilitated diffusion. That is diffusion of molecules that takes place from the higher concentration to a region of lower concentration. In simple diffusion, we don't require the carrier protein, but now I require a protein which is a selective protein which helps in transport of molecules. It can be by the channel proteins or the transporter proteins. So, what is the difference between channel proteins and transport protein? We'll have a discussion. But these are a types of facilitated diffusion. It can be uniport, symport, or antiport also. So, when you look about here, the channel proteins are simple proteins only. The channel proteins are simple protein. Just a minute, beta. Yeah. So we have channel protein and we have transport protein. In channel protein, the molecules will go along the concentration gradient, but only in one direction. Channel protein, they can go only in one direction. That is, it can enter inside or it can go outside. There's only one direction for the movement of molecules. In case of transport protein, there is an enzyme called permease, and that can go along the concentration gradient or they can go against the concentration gradient. This always can go along the concentration gradient. This can go along the concentration gradient, or using a membrane protein by utilizing the energy, they can go against the concentration gradient. Both of them are highly possible in case of transport protein. So channel protein, they what they do, only ions can move, and they does not require any energy. But transporter protein, when they're going in a downhill movement along the concentration gradient, there is no utilization of any energy. But they're going against the concentration gradient means they want the energy in the form of ATP. Along the concentration, there is no ATP utilization. In a nutshell, channel protein and transport protein are a type of protein which helps in transport of molecules. without using energy or with using energy, where channel protein they can go only one direction along the concentration gradient without utilizing energy in the form of ATP, whereas transporter protein they can allow the molecules to go in any direction, maybe along the concentration gradient where no ATP is utilized or against the concentration gradient. It's a nice graph which says about I'm increasing the solute concentration and rate of solute movement. I'm just checking here. When I'm able to see the rate of solute transfer means movement of molecules are occurring. In simple diffusion, you can see in this, in the given time, the rate of the solute movement is very, 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 very low. When I just take in green color, I'll take here. It's very low at the rate of facilitated diffusion. It is supporting the diffusion, so we can see they are nearly reached the mean max position here. Not even they able to meet 50% of them. So, which one is the better means of transport? I mean, all of them are better means of transport. But in case of passive movement, facilitator is the best means of transport because large number of molecules can be transported without using energy in the form of ATP. So, we are going to focus on this as an important means of transport of molecules compared to the simple diffusion. But osmosis is just a simple diffusion. But in case of movement of molecules in macro level, we require only protein mediated transport. So when we look about the protein mediated transport, that is called as a facilitated diffusion. For ions, there are some gated channels. Imagine that you have your wonderful home. In your home, you'll be having a door. So you have a calling bell, and when a person switches on the calling bell, you can check who is there. If it is a stranger, definitely you will not allow. If it is somewhat known person, you may ask them what and all, and you can send them there itself. It's little bit close means you can offer them a cup of tea, maybe in the hall itself. It's very friendly only. You can share everything to them. Same way, they are specific. The proteins are highly specific. They know which molecules has to pass through. So we have open or we have closed configuration are there, and we have some channel which are constantly open. They are called as pores. So if you look about here in this given example, these pores are continuously opened. But if you look about here, here they require a signal. So this gated channel is completely closed channel. So the closed channel cannot help in transport of molecules. But when a signal is given to them, the channels get open. The protein will modify the structure, the gate will get open. And when the gates get opened, the molecules are able to move from higher concentration to a region of lower concentration. As per the rule of osmosis, of course, you all know, the molecules always move from a region of higher concentration to a region of lower concentration through a semi permeable membrane in case of osmosis. But in this case, the molecules are able to move from a region of higher concentration to a region of lower concentration only by getting a signal in case of opened transport protein. Here, in case of closed transport protein, only after getting a signal they get opened. But but this is completely open type, and always they are remaining as open through a tiny pores. And there are different types of signals are there. Okay, no need to go too deep into the signal now. Only when a stimulus is given they get opened up. When there's no stimulus is given, they will not get opened up. For this example about voltage and ligand dependent, I'm going to take you one wonderful example is neurotransmitter impulses. Of course, the questions are different here. Propagation, you can see voltage and channels are there. But to understand this, let me tell you here. When you go with the transmission of impulse, this is called as the axon. The axon will be completely negatively charged. The inner core is completely negatively charged. And the outer area is completely positively charged, which is given over here minus minus. This condition is called as polarized. Now, when a stimulus is given, that is, we are giving a signal, That time what will occur? A voltage gated channels will get opened. That time what will happen? This was the initial condition. Now the charge will get reversed. The inner core will become positively charged. That is, the exoplasm will get positively charged. The outer ACF, they will become negatively charged. That is what is given here. This is called as depolarized. This potential is called as resting potential. This one is called as action potential. When the signal is given, so when the signal is given, then the following reaction takes place. The charge will get reversed. This is because of some selected channels are there. So when our selected channels are there, that is the movement of sodium and potassium movements are there. You'll be learning in depth, not now, in the days to come, about how the molecules are able to get transported. We can see sodium can move in, sodium can go up. So when sodium can move in, sodium can go up. So when the voltage gate channel gets opened, signal is given, channel enters in. When signal is off, Na plus goes out. So its influx and efflux of sodium is taking place. When you have doubt, when you come for the physical class or online class through communication, that time we can discuss more about that. Therefore, where and all it is highly functional means it is functional in your nerve conduction for the transmission of impulse, cell to cell contraction and sharing of informations, and in your receptors or for your hormonal actions for everything we require this signaling transport mechanism. So the only by that, even you I'm speaking to you, you're able to hear me. I'm scribbling something. You're able to see all this is because of some chemical channels activated in your body in your neuron. So I told you, porins. They are small pores which are always are in open state. These are called aquaporins. So when I look about the porins, these are the porins, which are nothing but a channel protein. If they allow movement of molecules only in one direction, ஒரே directionல மட்டும்தான் போக முடியும்னாக்கா, that is called uniport. If two molecules simultaneously, when they can go, it is called symport. Already I've discussed with that. But when two molecules can go in opposite direction through these porins, this is called as antiport. So we have three means: symport, or sorry, uniport, symport, and this one is called antiport. So the movement is brought by some aquaporins. So for discovering this no aquaporins, and Nobel Prize was shared in the year 2003. They have a permeable pores only for the movement of water molecules. This is why uniport, symport, and antiport, which are called co-transport. Uniport, the molecules can go in one direction. Symport, they can go in simultaneously. The two molecules can go simultaneously in the same direction. But in case of antipode, in antipode movement, they always can go only in antipode. They can go in both the directions. So this is how we have some of the transporter molecules. I told transporter molecule can go with any directions. So when you go with the transporter molecule, a solute have to bind with specific protein. This protein helps in transport of molecules by forming some conformational change in the proteinous molecule. So they have to make some change the shape. So when solute approaches, the protein have to change their shape and they have to transport the molecule outside. This one of the family is called as GLUT family transporters. It's called as GLUT. I'll tell you what is GLUT transporter. GLUT transporter is actually a protein. GLUT is actually a protein. It is called as glucose transmitter protein. Glucose transmitter protein. So you have to understand that when a solute or a molecule from an external source, if they have to cross the plasma membrane, they have to bind with a set of protein called as glut transporter protein. This is your GLUT. They will have some change in their shape, accept the molecule, and they passed towards the inner side. So this is called as influx. So influx of solute occurs in the following. Influx means inward movement. Have occurred in the following phenomena because of GLUT, that is glucose transporter protein. Now coming back to this, it's called active transport. So let us recall what is active transport once again. Active transport is nothing but the movement of molecules from a region of lower concentration to a region of higher concentration by utilizing energy. So if I just take some molecule here, the molecules are able to move with the help of some channel proteins only. So we have some channel proteins. This channel protein helps in the movement of molecules. Now these are the molecules. Here the molecules are more in number. So here the concentration of molecules are higher. Here the molecules are in lower concentration. Here the molecules move from a region of higher concentration to lower concentration in case of osmosis. But in case of active transport, the molecules always goes against the concentration gradient. They will go against the concentration gradient. That means what? They are going against the concentration gradient. Means for this, they require energy in the form of ATP. They require energy in the form of ATP, which will supply the energy. Using this energy, it is an upward movement, not a downward movement. It's upward movement going against the concentration gradient by utilizing energy in the form of ATP. Adenosine monophosphate joins with one molecule of inorganic phosphate to give you the adenosine diphosphate. Diphosphate joins with another molecule of inorganic phosphate to give you the ATP. This is the ATP cycle, the formation process. This is in energy releasing process. ATP will be broken down to adenosine diphosphate. So what energy is released? Inorganic phosphate. This is completely utilized for energy. So that's how we can see that ionic pumps are activated and by a rule called Nokia. I'll tell you what is Nokia. For the transport of molecule, Nokia 3Na2K plus rule. N stands for sodium. The sodium have to move outside the cell. K plus have to move inside the cell by utilizing energy in the form of ATP. So if I have a protein here, during this, using a channel proteins are there here, the 3Na plus molecule have to go outside. Whereas K plus, the two K plus molecules have to enter inside the cell. And for this process, Energy is utilized in the form of ATP. That's called 3Na plus 2K plus rules. So for this also we have a ATP binding protein cassettes are there and ionic pumps are also there, which undergoes the following reaction to transport the molecules from one concentration to another concentration, from a region of lower concentration to region of higher concentration, utilizing energy in the form of ATP. So that's what 3Na K plus rule for ATP production. So you can see three binding sites are always there for Na plus. You can remember the rule again. 3Na plus 2K plus rule. 3Na plus molecules are there, 2K plus molecule. Deox binding means three bindings. There is two binding sites are there for us. Three binding sites there for Na plus. So we have phosphorylation site where only ATP is utilized. So we can have the rule as Nokia. Na plus outside, how many? 3Na plus outside, 2K plus inside, utilizing energy in the form of ATP. As per the rule I have given here in the previous one, same rule can be applicable here. So this is how the same cycle which explained that how the transport of molecules. You can see for everywhere this is Na plus and this is K plus they are given here. So you can see here three Na plus molecules Nokia NO outside or inside. Na plus is able to move outside, but 2K plus is able to inside. Nokia. Na plus outside, K inside, and we have Nokia here. So this is how the transport of molecules takes place. So how the change will occur, the same thing. The 3Na plus 2K plus rule is given here. So active is always coupled with the ATP. So water, it may be an Na plus, it may be a K plus, it may be a Ca plus, it may be H plus. Water they coupled, the molecule is coupled always. That is, the molecule in the direction of its gradient and molecule against its gradient is always coupled. The rule is always anything that goes outside. For example, இங்க பாருங்க, if one molecule goes outside, other molecule have to get inside. Not the same molecule. Na plus, it's inside. Ca2 plus is outside. Additionally, Na plus is inside Naka, some other molecule. This is in symport transport. This is symport transport. Even they can go for antiport transport too. So active helps in transport of molecules by utilizing. Previously, when you have seen about glute, here it is actually called SGLT transport one. Now to teach you what is, I'll just give you expansion. The meaning is actually the same like what we discussed here. SGLT1. What is SGLT1? It is actually found in your body. It is for S stands for sodium, G stands for glucose, T stands for transport, L, it's a co-transport. What is L? Glucose. Sodium, glucose transporter, one protein. The same how we saw in glute, same way also. For glucose, we have a specific protein, they modify and they go for conformational changes and they push the molecule inside. The same meaning what I discussed in the previous one, in and out is determined by the conformation change of the proteins. So active transport and secondary transport when take into consideration, even for H plus also to control pH also we have Na and H or the Na and carbonate and chlorine transporters. So nothing to discuss more about that. This also we discuss about the membrane transport mechanisms. So let us summarize this chapter. Transport according to the concentration gradient. If they go along with the concentration gradient, is a passive transport. If they go against the concentration gradient by utilizing energy in the form of ATP, then it is called as active transport. And the various types of transport is simple and facilitated in case of passive. In active, they always utilize a carrier protein and they can utilize the energy in the form of ATP by the movement of ions. So I think it's done with our summary here. I think there should not be any doubt in the following topics what I discussed with you now. So in the next class, I'll be discussing about endocytosis and the exo. Now I think I have enough time. I'll discuss about endocytosis and exocytosis because recording only I can extend the time to. So we have endocytosis and we have exocytosis. Endo அப்படின்னா inside. Cytosis movement. Cellular movement. So inside movement and outside movement are also there. Okay. It takes some time for you to understand about endo and exo. I'll just give you here only one example, and next class I'll continue with the what is endo and exo. So we have an amoeba, and simple example I take here. If they have seen any foreign molecules, they'll form one feed called as pseudopodia, and next is they'll be completely engulfed intake, and one cavity will be formed here. One like a cavity means one vacuole will be formed here, and this is found to be the nucleus. Then what will occur here will be having the cell where one complete vacuolated structure will be formed, and this structure is called as phagosome. A cell eating phenomena is called as phagosome. So here the foreign molecules are taken inside. See the example, the foreign molecules are taken inside. This process is called as endocytosis. Exo means what? The same particle is thrown out. It's a process where the digestive particles are thrown out. This process is called as exocytosis. So we have endocytosis and exocytosis, and we have another one I'll be discussing a little bit later about an exocytosis and further molecular mechanism. Just give me some. So the endocytosis and exocytosis is a very big topic, though they are given in a simple way. So now I'll give you in a simple way. Next class I'll discuss in depth with you. Okay. So it's a mechanism where a foreign particle is taken in. Exocytosis is a mechanism where the foreign particles are or digestive particles are thrown out. இது மூலமா we have this diagram which will take nearly or 20 minutes to explain these two mechanisms. Anyway, this diagram makes it very clear about how the particles are taken in. Next class will continue with the membrane exchanges, endocytosis and exocytosis, which are very, very important topic, which will be taken for you on, okay, I'll record and send it to you whenever the time permits for me. Preferably on Wednesday, I'll record and send it to you. Okay. So that's all for today. The recording will be there with you, and have a good day better. Thank you, Ma.